there's all this problem with, self-diagnosis and self-reporting, and that just seems sort of intrinsic to the measures that you're using. So I just wondered if you could comment on sort of either methods that have to do with some of the social networking data that isn't conditioned on self-reporting, or perhaps neuroimaging studies, and whether some of these large studies that are taking place now, like the Connectome project, are going to have data in them for things like depression. I haven't worked on depression, but some of the other kinds of things are things that we're very interested in using neuroimaging because of the problems with self-reporting in other psychiatric diseases.
Yeah, it's for sure a big problem. I don't know that much on neuroimaging actually, but I think the one of the last slides I showed, that's a way of measuring, like measuring activity, social activity, also your voice, those kind of things. That's not self-assessment, right? So that those are symptoms that actually can be measured without the person himself. But the neuroimaging, I'm not very familiar with.
So you're not looking at that at all?
No.
Yeah. Okay, thanks.
Can I just comment on the depression issue? Is one that's been of interest because of the search for, the holy grail of biomarkers of depression.
Right.
And the one question is whether heart rate dynamics can add light on that, because there's sympathetic activation and vagal dysregulation. And there's some evidence we found that healthy middle-aged to older men have less complex heart rate dynamics at equivalent sleep stages. However, as a biomarker, it's probably going to be difficult. But the question I have was the idea of depression is it's not a disease, it's a syndrome with multiple causal factors. So peripartum depression and geriatric depression, people are both depressed. It's like the same endpoint, but as opposed to melanoma, which is is this extraordinarily elegant type of model condition to study. Depression medically is a name we give to multiple different routes to to some common endpoint. So I think it's going to be a challenge.
So can you use heart rate variability to measure changes in parasympathetic activity?
You can use them to assess that, but it's in terms of, it's not a diagnostic test in the sense of one person versus another, because you don't know, you need to know the person's baseline. But part of our interest in using, we would call it heart rate dynamics, or there's some dynamical space of different metrics of heart rate variability that can inform you about how that individual is relative to themselves. So you use the person as their own, in a sense, their own control, much in the spirit of what you've been looking at or embarking on here. But so many things confound the assessment of heart rate variability cross-sectionally: drugs, age, chronobiologic factors that were mentioned. So I think that one is going to be tough. But if you're comparing yourself with yourself and you want to see whether you've changed, then I think that's more hopeful.
So my question is maybe mostly for James, but probably the others. So those transitions in cellular systems that you see, like in chemically induced carcinogenesis, do you think they're driven by maybe like a specific mechanism, or do they resemble more like a phase transition where you have like very many different genomic states, like multiple combinations of mutations, maybe like even like almost uncountable?
Well, if you drew the simplest picture of a phase transition, it's basically two energy wells that pass through as you apply a constraint. Right. And so this is lower energy, then it switches to here, and there's a coexistence region in between. But the x-axis is a coordinate and has real live definition to it. And so right at the critical point, you can't interpret the x-axis because you've lost degrees of freedom and all. But as you head toward there, you can absolutely pull mechanistic information out, which is why these drug combinations work.
Yeah, but that's on the level that you observe. But like you referred to in your talk, you referred to not having to track all the genetic mutations, right?
Right.
But say, what if you had tracked them? Would you see that different cells have like completely different genomes? They just happen to lead to the same transition?
So we haven't done it here, although we're getting the data set. We've done it with a GBM case, very similar. It wasn't quite the same looking transition, but not that different. And in that case, we looked at the resistant tumor and the responsive tumor and the control tumor to see if there had been genetic. They were identical. We did not see, it was an adaptive response. It was not a genetic response. Now, for sure, one has Darwinian type responses, but in that case, the same cells that are responding to the drug, simply, the drug is like putting pressure on a balloon, The same air molecules in the balloon become solid if you put enough pressure on it. The cells that respond adapt and become resistant.
Thank you.
And I would point out that it's a particularly nefarious thing, because that adaptation can happen very quickly. You can have a drug that would be a great combination drug that would go in, fully engage the target, and you see no effect at all clinically because they have such a rapid adaptation. So
that happens more like on the signaling pathway level, not genetic.
yeah. So it happens with targeted inhibitors. That's what. Yeah, right.
You're going to deconstruct the entire NIH, the whole oncologic, mainstream. That's
Well, I think
it's remarkable because it's all about protocols and this, which is the real. It's quite something.
So I actually had a follow-up question to that because, for James again, you showed that phosphoprotein dynamics vary as you go from the melanoma to the neural crest states. What actual signaling pathways do these phosphorylations map onto? For instance, do we see migration pathways being affected, and is that the reason they're becoming more metastatic? So in terms of biological processes,
So we've just started doing expanded studies, and I can tell you that the transition is to a much more glycolytic phenotype. And so there is a very significant change in energy consumption that happens with this transition. These proteins are the ones that you normally would expect, like mTOR signaling, things like that. There's only a handful of candidates that you look at all the signaling pathways, there's a handful of candidates. But it's actually the connection to the metabolic pathways that I think is giving us deeper insight now.
right, because these phosphoproteins would map onto multiple pathways, right?
Oh,
And
yeah.
which ones of those are actually significant in manifesting the actual outcome of metastases or
if you
change
do a
in the cell
massive
state?
proteomic study, let's say you did 2,000 proteins and phosphoproteins from these cells, you would find that 1,700 of them are just devoted to the steady state of the cell. They have nothing to do with the cancer. They're just devoted to the cell just doing its regular thing, okay? And now you've got 300 that are devoted to the fact you've got a cancer cell. And now if you sample those 300, reasonably, 10 proteins or 20 that are really hitting different nodes in that network, you're actually capturing the essence of the cancer pretty well.
Yeah.
Single cell buys you a lot. You couldn't, at the bulk level, that's not true.
Couldn't
At the
look at
single
it.
cell level, it buys you a lot.
Yeah.
Okay, this is a question from Ingrid. So I know that you said something about emotional inertia predicting onset of depressive disorder. One thing I'm really curious about is SSRIs. One effect that people have with SSRIs is that it kind of flattens people out emotionally, meaning their highs are lower and their lows are higher. So I'm just curious, do SSRIs also, what exactly are the effects of SSRIs on fluctuation in auto-recorded emotions? Could they actually increase emotional inertia too?
What is an SSRI?
Oh, selective serotonin reuptake inhibitor, like Prozac and Paxil.
Oh, okay. And
Stuff they put in the water.
and then you're asking me how that affects the.
Yeah, how do those things affect fluctuations in auto-recorded emotions, especially since
a lot of depressive disorder is often associated with ruminations, and ruminations are often associated with this type of emotional inertia. Although SSRIs, they can help, reduce those ruminations, but on the other hand, there are also a lot of people who are on SSRIs also report that their emotions are still dulled, in the sense that their highs are lower and their lows, at least their lows are higher.
Okay, well, I have to admit, so I'm not a psychologist or a psychiatrist, right? So in that sense, I'm not sure. But what I do know is that these kind of things are actually targeting these connections in the network, right? So actually, you're trying to lower the connection strength to decrease feedbacks. So in that sense, it does. Completely affect the network itself. But I'm not sure if that's satisfying answer.
I'm not a psychiatrist either, but I slept in the Courtyard Marriott last night. So, but one thing as an MD I would just caution about with drugs is that their effects are very heterogeneous, and the same class of drugs in an individual, like with antiarrhythmics, can either make things better, not change them, make things worse. So the SSRIs, I don't know what the statistics are not. It's not a 100% effect. In fact, some people, don't think they work much at all, despite the initial responder reports. So I think it'd be hard to predict that. You might be able to say who's going to respond and who's not by how these other markers change. But those pharmacologic stuff is extremely difficult.
This one's for Dr. Goldberger. I'm curious, there's clearly a connection between breathing and heart rate variability, and my question is, there's a lot of emphasis out there in kind of the New Age community around different types of breath work and meditation, and I'm wondering, what's your sense as to whether these are helpful, harmful, both? Is there a danger with some of these practices, or are there some particular breathing practices that in fact increase resilience as measured by heart rate variability? So,
Can I take that? I was holding my breath while you were asking the question. The answer is that cardiopulmonary coupling, that the coupling between respiration and heart rate is extremely important, and that's the vagal tether is what connects them. And when you're young and fit, or if you're athletically trained, you'll have an enormous amount of what's called respiratory sinus arrhythmia. The heart rate goes up. You can feel it, or in a younger individual or an athlete. As people age, that's a biomarker of aging, is the loss of that autonomic modulation. The story with meditation is that you can actually, when people meditate, they breathe 4 or 5 times a minute, and they do get what we call exaggerated heart rate oscillations. Other people have reported that as well. Whether that has a sustained effect on integrative physiology and how to titrate that I don't think is known. Can you overdo it? Yeah, if you do it when you're driving, that's not recommended. The practice of meditation, has strong support, but there are people who are going to raise their eyebrows, levitate their eyebrows, and part of it is that there isn't the evidence there is not substantial. Obviously, if you meditated all day, that would be pathologic. So the question is, is 20 or 30 minutes? What's the prescription? How do you know in a given individual that you had a therapeutic effect? I think it's measurable. I think you can do it scientifically, but I don't think there's any strong data.
I guess my thinking is, when we think about wellness and health, if there were breathing techniques that could be taught to people that would in fact conduce to the kind of resilience.
So here's my response: that it's personalized medicine. That what may work for someone in terms of which variety of meditative thing do you do? Is it X or Y type of yoga? Is it golf? Is it fly fishing? What are you doing? And what's the physiologic measure? You're going to measure heart rate dynamics. But you can't any more than an SSRI is going to work in all depressed people. I would imagine that different types of meditation, there's a personal prescription. You need a way to follow that in an individual. The N is equal to 1. We don't have that, but I think it's it's doable. I think it's a great question, and I think it applies more broadly to everything we do. You can't just take it from the big data. This N is equal to 10,000, and then project it onto the individual. I think that's actually that's not good medicine. We do it all the time.
Is there any, if I want to do like a quantified self kind of thing, is there any good device?
I'm not licensed in the state to, I don't have a license in the state of Washington to make a prescription. I don't have an answer to that one. I don't think I'm not withholding, some deep truth. I just honestly don't know the answer.
Okay. So is there a difference between the situation where the agents in your system talk to each other and make all the global correlation that you see as critical point, or for example, or to the case that there is an external cause that makes in your traffic example, it would be a red light that makes all the cars stop, right?
No, in the traffic example, it's increased numbers of cars that makes the cars stop.
No, I mean the difference between these two situations. In one case, the traffic, because there are many cars, they talk to, the information is passed, you cannot go fast, so this causes the correlation. But if you have a red light and as an external cause, all the cars stop, there is again a correlation. Do you still call it a critical?
So there's a lot of the literature on the physics of phase transitions goes to that exact question. You apply something to the system, and what you're applying is basically influencing what are called constraints in the system, which are constraining the system to move in some direction, okay? But you don't always know what the connection is between the perturbation and the net influence. So if you apply, if you apply heat, it's temperature, okay? But if you apply a drug to cells, you don't know. You don't know. There are many constraints within the cell that'll respond, and it could differ from cell to cell. So you're asking a really sophisticated question, and it's not always easy to find out that cause and effect relationship directly. One can look at the global result, but the detailed pathway of that effect is very, very difficult to identify sometimes.
So the behaviors are almost similar, you say?
No.
It is
Behaviors would be very different. If you put a stoplight, there's no increased correlations. The cars just stop. And there's nothing to do with numbers of cars on the road. If you do it by traffic, you see divergent correlations that correlate with the numbers of cars on the road. So global observations are very, very different in those two cases.
Suppose you have the effect of an increased density of traffic. In one case, just due to the Lakers game is ended, and the other case because the LAPD has shut down a lane of one of those lovely things you have up there.
Yeah.
I used to live in San Diego. I never made it to LA. So those would look the same, wouldn't they? If you didn't, so intrinsically, or you could do it in different ways.
That's right. So I think the way you want to think of that's a good point. So when you take freshman chemistry and they teach you about pressure volume expansions, remember that? Okay. So you start off at pressure 1, volume 1, temperature 1, you end up at pressure 2, volume 2, temperature 2, okay? And out of this you can calculate changes in what are called state functions, free energy, things like this. But you can go from this state to this state in any one of about 8 million pathways, infinite number of pathways. No one ever asks about the pathways. In biology, they always ask about the pathways. They always ask about the pathways. But you can get lost, as biologists know, you can get lost in those pathways, okay? Because everybody can be different. State functions give you much more profound information about the system, much more robust.
If you consider the biological system as a nonlinear dynamic system, near the tipping point, besides your observer, a larger number of variables become highly, correlated. You have to. The other feature is they have to fluctuate this group for the number of the variables. So only observer correlation is not a sufficient condition. You have to consider other factors.
Okay, so this is a question for Dr. Goldberger. I know that you use dynamical assays to measure the heartbeat and glucose levels of individuals in order to figure out their entropy, fractality, and entropy. I'm just wondering, have you thought about applying this analysis for, say, brain waves, cortisol, or even maybe possibly the 700 metabolites and 400 proteins that Leroy Hood's plan measures in his 100K?
No. No, briefly. So the answer is that the notion of dynamical assays, of looking at the way things change in time as opposed to just taking the mean or the variance, that the time sequence, it's how you move, not just a little different. This different idea, but you have to have frequently sampled data of high resolution in a meaningful meaningful question. And the fact is that for most analytes, for most things that are taken out of blood, we're not even close. So you would need to continuously sample, say, cortisol, which would be interesting, and particularly related to depression. People have tried to look at cortisol in depression actually as a biomarker. The problem was they were measuring it twice a day after a dexamethasone suppression test and didn't pan out. But if they measured it every 5 minutes, there might be more information. So we have measured other things other than heart rate and glucose. We've looked at how people trace out a circle, how they make errors in the way they do a task. It turns out that the way you maintain kind of a homeostatic circle is to make micro errors that are correlated. We've looked at the way people maintain balance. EEGs, those signals are a mess. They're very, very difficult, and they're not at all like heartbeats. You got brain waves, that are sampled off the surface. You say, well, that's great. We can do, all these, apply all those metrical tests. Not so fast, because the signal is sort of a hodgepodge, and it's constantly changing state. That's much different than measuring the heartbeat. So I think one has to be attentive to the different signals you're looking at, and also there has to be something hypothesis driven about it, and then you have to do sort of the bioengineering. What are you sampling and how good is the accuracy? Those are very deep questions.
Maybe I just got the answer in terms of how frequently something needs to be measured, but is there some sort of low-hanging fruit that I can envision 5 years from now? I'm going to my doctor like 3 days in a row to get, or a blood test 3 days in a row to sort of get some sort of dynamic profile of something that could reveal something, or is that just not frequent enough?
Well, first of all, I think given the state of medical, just the sociology of medicine, the idea that you would be going to a doctor, with that frequency,
sure,
that the doctor would be sampling you.
Yeah, it's not, your annual physical turns into a multi-day testing type of thing to sort of pull out dynamics.
I think in terms of self-testing, we're in an era of everyone's measuring everything on themselves and getting big data, and then they throw out the intelligent watch or whatever it is, because no one knows what to do with the data. But our thought is not so much self-testing, but it would be really in the low-hanging fruit, as you put it, which has a lot of glucose in it, would be in assessing diabetic interactions, pharmacologic interactions with diabetes, to see what restored the complex dynamics, which drugs, added, you get an added dimension of insight. I don't see anything that, that in terms of self-testing or something that would go on in an MD's office in the very near term. But
Thanks.
prediction is a dicey business.
Okay, that'll be the last question.
Okay. So do we also see for insulin what we see for glucose?
Say that again?
Do we also see, for continuous insulin monitoring, do we see the same scene as we see in glucose?
Well, in our article on dynamical glucometry, we, it's just, sort of opening the field up. We said it would be interesting to have continuous, multiple channels, including insulin and glucose, to see, what the crosstalk was, but that's not available. Well, just for starters, it'd be nice to have more frequently sample glucose, which the monitors can do. They just don't report it because the physicians, are already overwhelmed.
Okay, please join us for the reception and the poster session. Many thanks to our speakers and thanks to everyone who stayed back.